Hello, my name is Patryk Dabek. I'm a junior at the Bergen County Academies, and today I'm going to be presenting about my research project, Exploiting Metabolic Reprogramming in Liver Cancer, Mitochondrial Membrane Potential Response to Selective ATP Hydrolysis Inhibition. So just to give a little bit of background information, my study deals with hepatocellular carcinoma, which is the most common type of liver cancer, and is the third most common cause of cancer-related death worldwide. One of the biggest complications in treating hepatocellular carcinoma are the underlying malignancies of the liver. The overwhelming majority of patients have some type of underlying cirrhosis of the liver, which can be caused by a variety of things, including viral hepatitis, cystic fibrosis, hemochromatosis, and many more conditions. In turn, these conditions make hepatocellular carcinoma so much more difficult to treat. This is because traditional therapies struggle to maintain post-treatment liver function. One of the hallmarks of cancer is metabolic reprogramming. In the presence of oxygen, normal cells would use oxidative phosphorylation to generate the maximum number of ATP. However, cancer cells use glycolysis, meaning that they generate less ATP. However, this serves a purpose. By utilizing glycolysis, they can shuttle metabolic intermediaries into the macromolecular biosynthesis pathways that allow cancer to proliferate so rapidly. Like all cells, cancer cells need to maintain their mitochondrial membrane potential to prevent cell death. Since the electron transport chain is no longer pumping protons into the intermembrane space, cancer cells need to find a different way to maintain this gradient. They are forced to use the reverse action of ATP synthase, ATP hydrolase, to maintain this gradient potential. Since ATP synthase is a dual action rotor, one direction of the enzyme can be selectively inhibited by having a small molecule bind to the rotor in a specific way. I first learned about this dual action mechanism when I was doing research for a presentation about heart attacks. I was trying to figure out why the nutrients in the heart cells were being used up so rapidly. After a bit of digging, I found a journal article that showed that under ischemic conditions, myocardial cells switch the direction of their ATP synthase, thus hydrolyzing ATP. After some more research, I found that there was a pharmaceutical company working on developing a drug that would selectively inhibit ATP hydrolysis for the purpose of stopping the depletion of nutrients in the myocardial cells. As previously mentioned, since cancer cells are reliant on ATP hydrolase to maintain their mitochondrial membrane potential and stay alive, I decided that this drug could be applicable as a cancer treatment. Therefore, I wanted to study the effects of ATP hydrolase inhibition on cellular metabolism, mitochondrial membrane potential, and cellular proliferation. I hypothesized that if ATP hydrolysis was inhibited, then the mitochondrial membrane potential would in turn collapse, releasing pro-apoptotic proteins into the cytosol, inducing apoptosis, and targeting the growth-supportive microenvironment of cancer tumors. Fundamentally, this study sought to demonstrate and investigate the ability of selective inhibition of ATP hydrolase to target mitochondrial membrane potential and decrease cell proliferation. I want to go on and talk about some of the assumptions of this study. So my experiment uses an in vitro model using the HepG2 cell line as a representative for hepatocellular carcinoma. These cells serve as a good representation of real human liver cancer because they hold a lot of the same phenotypes as cancer holds in patients in the real world. Furthermore, this cell line has been proven through multiple studies as an excellent representation of liver metabolism and environmental cytotoxicity. Another assumption was made in this study's analysis of cell death. I wanted to focus on the mitochondrial permeability transition port pathway since it is dependent on mitochondrial membrane potential and its depolarization leads to the output of pro-apoptotic proteins. This narrowed analysis allows for the examination of the role of mitochondrial membrane potential on cell death. I've been lucky enough to be able to conduct my research at my high school in the Nano-Structural imaging lab with my mentors Ms. Waldron and Dr. Reeves. This has allowed me to work with a lot of high-level equipment and practice my technique on a variety of different assays. On the day-to-day, a lot of what I use is just cell culture equipment, so I have my HepG2 cells from the American type culture collection, which I feed with RPMI 1640 medium with 10% FBS and 1% pen strep. Furthermore, I have access to a variety of equipment that allows me to attempt a variety of different assays. I've gotten to use a flow cytometer, a laser scanning confocal microscope. I've been able to attempt ELISAs and work on a bunch of different assays that can be read through a plate reader. One of the procedures that I conducted often in the lab was subcellular fractionation. I use this to gain fractions of mitochondria and cytosol. That way I can examine them and see the kinetics of different proteins as they move from the mitochondria to the cytosol. I would grow cells in T75 flasks until they were about 75% confluent, and then I would treat them for 24 hours. After that, I would harvest them and pellet them down using a centrifuge. From there, I would use a variety of different buffers and centrifugation techniques to separate the different cell fractions to gain what I needed to conduct my experiments. I also worked on enzyme-linked immunosorbent assays, or ELISAs, in which the expression of a protein could be measured by looking at the bindings of antibodies to that antigen and then analyzing the colorimetric changes that occurred depending on the concentration of the antigen in the sample. Feel free to pause on this slide that goes more into depth on the procedures that I used, but now I'm going to be talking about the results that I had. To examine the metabolic changes that occurred in cancer cells after treatment, I looked at glucose uptake and differences in ATP levels. These both decreased, showcasing that cells became less metabolically active and that ATP was being hydrolyzed inside of the cells. Furthermore, I conducted a flow cytometry experiment to look at the effect of treatment on cell cycle, and it was found that selective ATP hydrolysis inhibition actually induced a GOG1 cell cycle arrest, even though the other controls did not, meaning that this treatment prohibits cells from proliferating rapidly. To analyze the effect of treatment on cellular proliferation and viability, I conducted a MTS assay which uses a tetrazolium salt to quantitatively deduce if cellular mitochondria are active. Over 24, 48, and 72 hours, selective ATP hydrolase inhibition did in fact lead to a decrease in mitochondrial activity. Additionally, cell death increased based on a trypan blue exclusion assay and an increase in reactive oxygen species production. All this data tells us that in fact the cancer cells are entering apoptosis, but now we need to figure out why. The mitochondrial permeability transition pore works by opening the voltage-dependent anion channel when the mitochondrial membrane potential collapses. This induces the release of cytochrome C, which recruits APAF1 and procaspase 9, forming the apoptosome and leading to the activation of the executioner caspases 3 and 7, thus inducing apoptosis. To qualitatively determine mitochondrial permeability transition pore opening, we need to look at cytochrome C levels in the mitochondria and the cytosol. Through the use of subcellular fractionations, we can determine the kinetics of cytochrome C. Looking at the graph on the left, we can see that as soon as cells are treated, cytochrome C levels increase in the cytosol. This corresponds to an increase in the executioner caspase levels, meaning that in fact the depolarization of mitochondrial membrane potential is leading to the activation of apoptosis. Since the binding of CMX rosamine is dependent on the polarization of the mitochondria, we can use fluorescence to determine if the mitochondria have been depolarized. Looking at flow cytometry data in comparison to rotenone, a pesticide that kills all cells, and DMSO control, only ATP hydrolase inhibition treatment created a collapse of mitochondrial membrane potential. This was further confirmed by examining fluorescence of cells under a fluorescent microscope, as seen on the pictures on the right. To analyze these pictures, I used Fiji and ImageJ software to isolate the red color channel and measure the fluorescence of CMX rosamine in the cells. Furthermore, statistical data analysis was performed on all of my assays to determine if they were statistically significant by using a t-test in Excel. I've also attached annotated diagrams throughout this presentation to help better describe my research. To sum up my results looking at the metabolism and cellular proliferation data, we can see that the cancer cells became less active and proliferated less. Furthermore, looking at the data from the mitochondrial permeability transition pore and that of the mitochondrial membrane potential, we can see that as the membrane collapsed, pro-apoptotic proteins entered the cytosol, causing the cell to enter apoptosis. Ultimately, this study showcases that ATP hydrolysis inhibition can cause apoptosis in cancer cells without affecting ATP synthase, thus making it a prospective target for clinical usage as it wouldn't affect normal cells. This can allow for the development of a variety of different real-world therapies, such as pharmaceutical moieties, which can selectively target cancer cells to kill them. In the future, I hope to adapt my model to deal with more of the distinct genomic and clinical characteristics that cancer condones. Furthermore, I hope to involve more microscopy techniques to get a better understanding of changes in mitochondrial morphology. Additionally, I hope to use computational analysis since it can be used to better predict patient outcomes. I would like to thank the Bergen County Academies for providing me with the tools and resources to pursue my research, and I would also like to thank my mentor, Ms. Waldron, for always supporting me throughout my research process. Lastly, I would like to thank you, the listener, for listening to this presentation, and I hope you took something away from my study. Thank you.